Tonight's lecture is over Chapter 7, Cellular Division, and the first part of this lecture we're going to discuss mitosis. So we'll first discuss what a cell accomplishes when it reproduces. When you hear the word reproduction, you probably think of the birth of a new organism, but reproduction actually occurs more often at the cellular level. Consider the skin on your arm. Skin cells are constantly reproducing themselves and moving outward toward the surface, replacing dead cells that have rubbed off. This renewal of your skin goes on throughout your life, and when your skin is injured, additional cell reproduction helps heal the wound. When a cell undergoes reproduction or cell division, the two daughter cells that result are genetically identical to each other and to the original parent cell. Biologists traditionally use the word daughter in this context. It does not imply gender. Before the parent cell splits into two, it duplicates its chromosomes, the structures that contain most of the organism's DNA. Then, during the cell division process, one set of chromosomes is distributed to each daughter cell. As a rule, the daughter cells receive identical sets of chromosomes. Cell division plays several important roles in the lives of organisms. For example, within your body, millions of cells must divide every second to replace damaged or lost cells. Another function of cell division is growth. All of the trillions of cells in your body are the result of repeated cell division that began in your mother's body with a single fertilized egg cell. Another vital function of cell division is reproduction of the organism. Single-celled organisms, such as amoebas, reproduce by dividing in half, and the offspring are genetic replicas of the parent. Because it does not involve fertilization of an egg by a sperm, this type of reproduction is called asexual reproduction. Offspring produced by asexual reproduction inherit all of their chromosomes from a single parent. Many of the multicellular organisms can reproduce asexually as well. For example, some sea star species have the ability to grow new individuals from fragmented pieces. And if you've ever grown a house plant from a clipping, you've observed asexual reproduction in plants. In asexual reproduction, there is one simple principle of inheritance. The lone parent and each of its offspring have identical genes. The type of cell division responsible for asexual reproduction and for the growth and maintenance of multicellular organisms is called mitosis. Sexual reproduction is different. It requires the fertilization of an egg by a sperm. The production of the egg and the sperm cells involves a special type of cell division called meiosis, which occurs only in the reproductive organs, such as the testes and the ovaries in humans. We will discuss meiosis in detail in a subsequent video. And, just for reference, a sperm or egg cell has only half as many chromosomes as the parent cell that gave rise to it. So the gist of it is that there are two kinds of cell division involved in the lives of sexually reproducing organisms. Meiosis for reproduction and mitosis for growth and maintenance. So in order for us to understand both mitosis and meiosis, we must first discuss DNA, or deoxyribonucleic acid. DNA is the genetic material of living organisms. In humans, DNA is found in almost all cells of the body and provides the instructions that they need to grow, function, and respond to their environment. When a cell of the body divides, it will pass on a copy of its DNA to each of its daughter cells. DNA is also passed on at the level of organisms. When the DNA in a sperm cell meets the DNA of an egg cell, and they combine to form a new organism that has the genetic material from both parents. Physically speaking, DNA is a long string of paired chemical units that we call nucleotides that come in 4 different types. And it also carries information organized into units called genes. Genes typically provide the instructions for making proteins, which give cells and organisms their functional characteristics. In eukaryotic organisms, such as plants and animals, the great majority of DNA is found in the nucleus and is called nuclear DNA. In bacteria and other prokaryotes, most of the DNA is found in a central region called the nucleoid, which functions similarly to a nucleus but is not surrounded by a membrane. So remember that bacteria and other prokaryotic organisms lack membrane-bound organelles, which distinguish them from eukaryotic organisms. A cell's set of DNA is called its genome. Since all of the cells in an organism, with only a few exceptions, contain the same DNA, you can also say that an organism has its own genome, and since the members of a species typically have similar genomes, you can also describe the genome of a species. In general, when people refer to the human genome or any other eukaryotic genome, they mean the set of DNA found in the nucleus, that is, the nuclear genome. Moving on, each species has its own characteristic number of chromosomes. For instance, humans have 46 chromosomes, bees have 32 chromosomes, and dogs have 78 chromosomes. Like many species of animals and plants, humans are diploid, meaning that most of their chromosomes come in matched sets known as homologous pairs. Thus, the 46 chromosomes of a human cell are organized into 23 pairs, and the two members of each pair are said to be homologs of one another, with the exception of the X and Y chromosomes. The X and Y chromosomes are said to be hemizygous. In contrast, human sperm and egg cells, which have only one homologous chromosome from each pair, are said to be haploid. When a sperm and egg fuse, their genetic material combines to form one complete diploid set of chromosomes. So for each homologous pair of chromosomes in your genome, one of the homologs is maternally inherited and the other is paternally inherited. Moving on, so there are 23 pairs of chromosomes in humans for a total of 46 chromosomes in a normal somatic cell. And there are two types of chromosomes. There's autosomes. Autosomes are any chromosome that is not the sex chromosome. And the other type of chromosome is the sex chromosome, which is made up from either your X or your Y chromosomes. Females are going to have two X chromosomes, and males are going to have an X and a Y chromosome. And in the human, the haploid cells, which are your sperm and eggs, have only 23 chromosomes total. I believe that this haploid number in your germ cells contributes to sex drive, in the respect that since they have half the amount of DNA that a somatic cell would have, the sex cells or the germ cells need to find another cell to make themselves whole. It's a hypothesis of mine. This next slide shows what we call a karyotype. So the two chromosomes in a homologous pair are generally very similar to one another. They're the same size and shape and have the same pattern of light and dark bands. Bands appear when the chromosomes are stained with a dye, and the dark bands mark more compacted DNA, while the light bands mark less compacted DNA. Most importantly, the two homologs in a pair carry the same type of genetic information. For instance, there is a gene found near the bottom of chromosome 15 that affects eye color. A person might have the blue version or allele of this gene on one homolog, but the brown version on another. Both homologs have the same type of gene in the same place, but they can, and often do, have different versions of genes. Furthermore, in humans, the X and Y chromosomes determine a person's biological sex. As I've already stated, females will have two X chromosomes, and males will have an X and a Y chromosome. While the two chromosomes in a woman's cells are genuinely homologous, The X and Y chromosomes of a man's cells are not. As I stated earlier, we call that hemizygous. They differ in size and shape, with the X being much bigger than the Y. Actually, the Y chromosome is the smallest chromosome in the human genome. So we can say that women have more DNA than men do. Furthermore, the X and the Y chromosome contain mostly different genes. The X and the Y chromosomes are known as sex chromosomes, and the other 44 chromosomes are called autosomes. And that brings us to chromosomal structure and compaction. Each eukaryotic chromosome contains one very long DNA molecule, typically bearing thousands of genes. The number of chromosomes in a eukaryotic cell depends on the species. Chromosomes are made up of a material called chromatin, which is a combination of DNA and protein molecules. The protein molecules help to organize the chromatin and help control the activity of its genes. Most of the time, the chromosomes exist as a diffuse mass of fibers that are much longer than the nucleus they are stored in. In fact, if stretched out, the DNA in just one of your cells would be taller than you. As a cell prepares to divide, its chromatin fibers coil up, forming compact chromosomes. When they are in this state, chromosomes are clearly visible under a light microscope. When a cell is not dividing, the chromosomes are too thin to be seen under a light microscope. Such long molecules of DNA can fit into the tiny nucleus because within each chromosome, the DNA is packed into an elaborate, multi-level system of coiling and folding. A crucial aspect of DNA packaging is the association of DNA with small proteins called histones found only in eukaryotes. Bacteria do have similar proteins, but prokaryotes lack the degree of DNA packaging found in eukaryotes. So the figure on the left hand of the slide presents a simplified model for the main levels of DNA packaging. At the first level of packaging, the histones attach to the DNA. In electron micrographs, the combination of DNA and histones has the appearance of beads on a string. Each quote-unquote bead is called a nucleosome, and this consists of DNA wound around histone molecules. When not dividing, the DNA of active genes takes on this lightly packed quote-unquote beads on a string arrangement. When preparing to divide, chromosomes pack further. At the next level of packaging, the beaded string is wrapped into a tight helical fiber. This fiber coils further into a thick supercoil. Looping and folding can further compact the DNA, as you can see in the chromosome at the bottom of the figure. Viewed as a whole, this figure gives a sense of how successive levels of coiling and folding enable a huge amount of DNA to fit into a cell's tiny nucleus. Before a cell begins the division process, it duplicates all of its chromosomes. The DNA molecule of each chromosome is copied through the process of DNA replication, and new protein molecules attach as needed. The result is that each chromosome now consists of two copies called sister chromatids, which contain identical genes. The two sister chromatids are joined together tightly at a narrow waist called the centromere. When the cell divides, the sister chromatids of a duplicated chromosome separate from each other. Once separated from its sister, each chromatid is considered a full-fledged chromosome, and it is identical to the original chromosome. One of the new chromosomes goes to one daughter cell, and the other goes to the other daughter cell. In this way, each daughter cell receives a complete and identical set of chromosomes. A dividing human skin cell, for example, has 46 duplicated chromosomes, and each of the two daughter cells that result from it has 46 single chromosomes. Moving on, how does chromosome duplication and cell division fit into the life of a cell? The rate at which a cell divides depends on its role within the organism's body. Some cells divide once a day, others less often, and highly specialized cells, such as mature muscle cells, not at all. The ordered sequence of events that extends from the time a cell is first formed from a dividing parent cell until its own division into two cells is called the cell cycle. Most of the cell cycle is spent in interphase. This is a time when a cell performs its normal functions within the organism. For example, a cell in your stomach lining might make and release enzyme molecules that aid in digestion. During interphase, a cell roughly doubles everything in its cytoplasm. It increases its supply of proteins, increases the number of many of its organelles, such as mitochondria and ribosomes, and it grows in size. Typically, interphase lasts for at least 90% of the cell cycle. So from that standpoint of cell reproduction, the most important event of interphase is chromosome duplication, when the DNA in the nucleus is precisely doubled. This occurs approximately in the middle of interphase, and in the period when it's occurring is called the S phase. That stands for DNA synthesis. The S is for synthesis. The interphase periods before and after the S phase are called the G1 and G2 phases, respectively. G stands for gap. During gap 2, or G2, each chromosome in the cell consists of two identical sister chromatids, and the cell is preparing to divide. The part of the cell cycle when the cell is actually dividing is called the mitotic phase, or the M phase. It includes two overlapping processes, mitosis and cytokinesis. In mitosis, the nucleus and its contents, notably the duplicated chromosomes, divide and are evenly distributed, forming two daughter nuclei. In cytokinesis, the cytoplasm is divided into two. Cytokinesis usually begins before mitosis is completed. The combination of mitosis and cytokinesis produces two genetically identical daughter cells, each with a single nucleus surrounding the cytoplasm with organelles and a plasma membrane. So let's move on to interphase. Interphase is the period of cell growth when the cell makes new molecules and organelles. Within the nucleus, the chromosomes are duplicated, but they cannot be distinguished individually because they are still in the form of loosely packed chromatin fibers. Moving on to mitosis, this cartoon on the right hand of your slide illustrates the cell cycle for an animal cell. Mitosis is a continuum, but biologists distinguish four main stages: prophase, metaphase, anaphase, and telophase. The chromosomes are the stars of the mitotic drama, and their movements depend on the mitotic spindle, which is a football-shaped structure of microtubules that guides the separation of the two sets of daughter chromosomes. The spindle microtubules grow from two centrosomes, which are clouds of cytoplasmic material that in animal cells contain centrioles. Centrioles are can-shaped structures made of microtubules. Cytokinesis is the division of the cytoplasm into two genetically identical daughter cells, and this typically occurs during telophase. In animal cells, the cytokinesis process is known as cleavage. The first sign of cleavage is the appearance of what we call a cleavage furrow. This is an indentation at the equator of the cell. A ring of microfilaments in the cytoplasm, just under the plasma membrane, contracts like the pulling of a drawstring, deepening the furrow and pinching the parent cell in two. In contrast, cytokinesis in a plant cell occurs differently. Membranous vesicles containing cell wall material collect at the middle of the cell. The vesicles gradually fuse, forming a membranous disc called the cell plate. The cell plate grows outward, accumulating more cell wall material as more vesicles join it. Eventually, the membrane of the cell plate fuses with the plasma membrane, and the cell plate's contents join the parental cell wall. The result is two daughter cells. So let's move on and detail the steps of mitosis. During prophase, changes occur in both nucleus and cytoplasm. In the nucleus, the chromatin fibers coil so that the chromosome becomes thick enough to be seen individually with a light microscope. Each chromosome appears as two identical sister chromatids joined together at the narrow quote-unquote waist of the centromere. In the cytoplasm, the mitotic spindle begins to form as microtubules grow out from the centrosomes. The centrosomes are moving away from each other. Late in prophase, the nuclear envelope breaks up. The spindle microtubules attach to the centromeres of the chromosome and move the chromosomes towards the center of the cell. And in this slide, we have a more detailed description of prophase along with the cartoon. So we see that the nuclear envelope breaks down, the centrosomes move to opposite poles of the cell, they send out microtubules and form the mitotic spindle. Then the sister chromatids begin to coil and become more tightly condensed. Now they are visible under a light microscope, and the kinetochore forms at the centromere. Once the nuclear envelope is fully broken down, the mitotic spindles are attached to the centromere from both centrosomes. And that brings us to metaphase. In metaphase, the mitotic spindle is now fully formed. The centromeres of all the chromosomes line up on an imaginary plate between the two poles of the spindle. For each chromosome, the spindle microtubules attached to the two sister chromatids pull toward opposite poles. This tug of war keeps the chromosomes in the middle of the cell. And that brings us to anaphase. Anaphase begins suddenly when the sister chromatids of each chromosome separate. Each is now considered a full-fledged daughter chromosome. The chromosomes move toward the opposite poles of the cell as the spindle microtubules shorten. Simultaneously, the microtubules that are not attached to the chromosomes lengthen, and this pushes the poles farther apart, and the elongation of the cell begins to occur. Telophase begins when the two groups of chromosomes have reached opposite ends of the cell. Telophase is the reverse of prophase. Nuclear envelopes form, the chromosomes uncoil, and the spindle disappears. Mitosis, the division of one nucleus into two genetically identical daughter nuclei, is now finished. Cytokinesis, which is the division of the cytoplasm, usually occurs with telophase. In animal cells, a cleavage furrow pinches the cell in two, and this produces two daughter cells. And in plant cells, a membranous disc called the cell plate forms, and it grows outward and eventually fuses with the plasma membrane of the cell. So this slide just shows us an overview of the process of mitosis in cartoon form. We see at about 12 o'clock of the slide, we have a single parent cell, and that leads into prophase. Then metaphase begins, then anaphase, and then telophase, then cytokinesis, and the result is two daughter cells. Moving on to cell cycle checkpoints. For a plant or animal cell to grow and maintain its tissues normally, it must be able to control the timing of cell division. The sequential events of the cell cycle are directed by a cell cycle control system that consists of specialized proteins within the cell. These proteins integrate information from the environment and from other body cells to send a quote-unquote stop or a quote-unquote go-ahead signal at certain key points during the cell cycle using transduction pathways. For example, the The cell cycle normally halts within the G1 phase of interphase unless the cell receives a go-ahead signal via certain cell cycle control proteins. If that signal never arrives, the cell will switch into a permanently non-dividing state. For example, some of our nerve cells and muscle cells are arrested this way. If the go-ahead signal is received and the G1 checkpoint is passed, the cell will usually complete the rest of the cell cycle. And that brings us to cancer. The reason we discuss cancer and cell division is because cancer is a disease of the cell cycle. Cancer cells do not respond normally to the cell cycle control systems. They divide excessively and can invade other tissues of the body. If unchecked, cancer cells may continue to divide until they kill the host. The abnormal behavior of cancer cells begins when a single cell undergoes transformation, a process whereby a normal cell converts to a cancer cell. Transformation occurs after a genetic change or a mutation in one or more genes that encode for proteins in the cell cycle control system. Because a transformed cell grows abnormally, the immune system normally recognizes and destroys it. However, if the cell evades destruction, it may proliferate to form a tumor, an abnormally growing mass of the body cells. If the abnormal cells remain at the original site, the lump is called a benign tumor. Benign tumors can cause problems if they grow large and disrupt certain organs, such as the brain, but often can be completely removed by surgery. In contrast, a malignant tumor can spread into the neighboring tissues and other parts of the body, displacing normal tissue and interrupting organ function. An individual with the malignant tumor is said to have cancer. Cancer cells may separate from the original tumor or secrete signal molecules that cause blood vessels to grow toward the tumor. A few tumor cells may then enter the circulatory or lymphatic system and move to other parts of the body where they may proliferate and form new tumors. The spread of cancer cells beyond their original site is called metastasis. Blood vessels growing toward the tumor is called angiogenesis. Cancers are named according to where they originate. Liver cancer, for example, always begins in the liver tissue and may spread from there. Once a tumor starts growing in the body, how can it be treated? The three main types of cancer treatment are sometimes referred to as slash, burn, and poison. Surgery to remove a tumor, slash, is usually the first step. Burn and poison refer to the treatments that attempt to stop cancer cells from dividing. In radiation therapy, burn, parts of the body that have cancerous tumors are exposed to concentrated beams of high-energy radiation, which can often destroy cancer cells without seriously injuring the normal cells of the body. However, there is sometimes enough damage to normal body cells to produce unwanted side effects such as nausea and hair loss. Chemotherapy, or poison, uses the same basic strategy as radiation. In this case, drugs are administered that disrupt cell division. These drugs work in a variety of ways. Some prevent cell division by interfering with the mitotic spindle. For example, paclitaxel or Taxol freezes the spindle after it forms, keeping it from functioning. Taxol is made from a chemical found in the bark of the Pacific yew, a tree found mainly in the northwestern United States. It has fewer side effects than many other anti-cancer drugs and seems to be effective against the hard-to-treat cancers, such as the cancer of the ovary and the cancer of the breast. Another drug called vinblastine prevents the mitotic spindle from forming in the first place. Vinblastine was first obtained from the periwinkle plant, which is native to the tropical rainforests of Madagascar. Although cancer can strike anyone, there are certain lifestyle changes that you can make to reduce your chances of developing cancer or increase your chances of surviving it. For instance, not smoking, exercising adequately, Avoiding overexposure to the sun and eating a high-fiber, low-fat diet can help reduce the likelihood of getting cancer. 7 types of cancer can be easily detected: skin and oral via physical exam, breast via self-exams and mammograms for higher-risk women, prostate via rectal exam, cervical via pap smear, testicular via self-exam, and colon via colonoscopy. Regular visits to the doctor can help identify tumors early, which is the best way to increase the chances of successful treatments regarding cancer. So let's summarize and recap tonight's lecture. After tonight's lecture, you should know what an organism's genome is. You should know what are the 2 states of chromosome numbers, and what are the 2 types of chromosomes in humans. You should be able to list the important layers of chromosome structure. You should know the 3 phases of interphase. You should also know when sister chromatids form. You should know what is the order of the phases of mitosis. You should also know what events happen in each phase of mitosis. You should be able to draw out the stages of mitosis. You should know the difference between cytokinesis in animals and plants. You should know where cell division regulations come from, and you should know some examples of those. You should know where the cell cycle checkpoints are, and you should know how genes involved in cell division relate to cancer.